Hello everyone and welcome to the biology lecture series, and today we'll be discussing the topic heredity and the inheritance. So in the previous lecture we were studying about the Mendel's experiment. We studied about the terms which are related to the genetics. So we studied about the dihybrid cross, monohybrid cross, trihybrid cross, and we also studied about the back cross and the test cross. And then we also studied about the other terms such as homozygous individual, heterozygous individual. All right. So now this lecture is the continuation of the previous lecture. And in this section, we'll study again about the Mendel's experiment. Okay. So let's start with this lecture. So Mendel, he was working on the garden pea. So Mendel chose garden pea as a plant. So why he chose this plant only? Why only garden plant only? Garden pea only. Alright, so these are the reasons why he chose the garden pea plant for carrying out the experiments. First of all, first reason is it has well-defined characters. Clear? So all the traits, all the characters, they were very well defined, and you can see them in the phenotype. Okay, if you look at the morphology, you can see, okay, the flowers are yellow colored or the flower are some other colored. Okay, you can also see the shape of the seed, whether the seed is round shape or it is wrinkle shaped. Okay, so all the characters, they were very well defined. Clear? They were a clear distinction. You can also say there was a very clear distinction between the characters. Okay, and the traits were contrasting. Okay, contrasting traits were present. Contrasting traits were present. Clear? Contrasting trait means one of the traits, if we talk about the height. All right, if one trait is height, then you can say the plant was tall or it was dwarf. Okay, so characters are very well defined. It is tall or it is dwarf. There is no intermediate stage here. Clear? Again, if we talk about the shape of the seed. Okay, if you talk about shape of the seed, okay, it can be round or it can be wrinkled. Clear? So you can see that the characters are very well defined and contrasting characters or contrasting traits were present. Clear? Next is bisexual flowers were present. Okay, bisexual flowers means, okay, the flower which consists of both the female as well as the male reproductive system or the parts. Clear? This is known as the bisexual flowers. Clear? So in bisexual flowers, self-pollination will be promoted. Clear? Now, these plants were predominantly self-fertilizing. Clear? Because they had the bisexual flowers. Bisexual flowers are what flowers? They contain both the types of the reproductive part. Okay? Both the type of reproductive part means male as well as the female. That's why they are predominantly self-fertilizing. Okay, self-fertilization means the anther, the pollens from the anther of the flower can land on the stigma, okay, of the same flower. Or you can say transfer of the pollen from the anther to the stigma of the same flower. This is known as fertilization. Now this pea plant, it was very easy to hybridize. Okay, so hybridization was very easy in pea plant. So because of this hybridization, hybrids are produced. Clear? Now let's talk about the crossing technique employed by the Mendel. So how did Mendel do the crossing in this pea plant? Clear? What technique or what steps he employed for the crossing? Now, he knows since the garden pea is self-fertilizing. Clear? Means it can undergo self-fertilization, means the pollens of the anther can, you can say, land on the stigma of the same flower. Okay, the anthers have to be removed before they mature. All right. Now, you know, the pea plant, they are predominantly self-fertilizing because they have bisexual flowers. So what he had to do, he had to remove the anthers, okay, before they get mature. If they get mature, they will release the pollen grains. So emasculation was done. Emasculation is the removal of the anthers before they mature. So this will promote the cross pollination. All right. So first of all, emasculation was done. Then what happens? The stigma is protected against any foreign pollen with the help of a bag. So bagging is done. So stigma is protected against any foreign pollen with the help of a bag. Clear? The pollens then at the dehiscence stage is brought from the plant to be used as a male plant, and it dusted on the stigma of the emasculated flower. Clear? So initially, what was the first step? Emasculation. In second step, what was done? The cross pollination was prevented. Okay, so we want a specific pollen to land on the stigma that we have selected. All right, but we don't want any foreign pollen to land on the stigma. So we decide which pollen should land on the stigma. Okay, we selected. Okay, that's why the stigma is bagged. All right, so as to protect it against any foreign pollen. Clear? So this was the second stage. Now in the third stage, what was done? The pollens, okay, from the plant that we have selected, okay, it is dusted on the stigma, which is feathery. So you can say feathery stigma, all right, of the emasculated flower. So this is the third step. Now, okay, this is the third step. Fourth step, at the time of the pollination, the pollens should be mature and the stigma should be receptive. Clear? So this is the condition. All right. So the pollens should be mature when they land on the stigma. Okay. So stigma should be receptive and the pollen should be mature. All right. Whenever the pollen lands on the stigma. Clear? So stigma is the receptive part, means it receives the pollen. If the pollens are not mature, then the pollens will not germinate and it will not form the pollen tube. And the pollen will be incompatible. So for the pollen to be compatible, the pollen should be mature and the stigma should be receptive. So this is the condition here. So here you can see what was the crossing technique employed by the Mendel. First of all, he did the emasculation. Then he covered the stigma with the bag, such as a polythene bag. This is known as the bagging. All right, this was done to prevent the contamination from the foreign pollen. And what he did in the third step, we selected a particular pollen. So that a desired pollen from the desired plant, okay, that we have selected to be the male parent, okay, it was dusted on the stigma of the emasculated flower. Clear? And that's how pollination was done, or the crossing was done. Clear? Now these were the steps. First was the emasculation, all right, which is the removal of the anthers. Okay, so this is removal of the anthers. Then next is the bagging. Okay, bagging is done for the protection of the stigma, okay, from the foreign pollens. Clear? And then third is the dehiscence. Dehiscence was dusting the pollens, okay, from the desired plant. This is clear that we have selected to be the male parent. Okay, that will be the male parent. Alright, and the pollination was done. Clear? So these were the technique or the crossing technique employed by the Mendel. Now the traits chosen by the Mendel for the experiment. So there were 7 traits that were chosen by the Mendel to carry out its experiment. So you can see the traits are very well defined and they are contrasting also. First was the stem height. Clear? So you can see stem height, either the plant was tall, so if for tall, then the dominant. So dominant character here is the tall, and the recessive character or the recessive trait is the dwarfness. Then next is the flower color. So violet is the dominant, okay, flower color, and white is the recessive flower color. Next is the flower position. So flower can be axially located or terminally located. Clear? Axially means in the axis. All right, and second one is the terminal of the shoot, you can say, at the terminal positions. Next is the pod shape. So it can be inflated or it can be constricted. And then we have the pod color. Pod color can be green or it can be yellow. Then seed shape. Okay, it can be round or it can be wrinkled. Then seed color, it can be yellow or it can be green. Clear? So these were the 7 pair of contrasting characters. So you can also say these are the 7 pairs of contrasting characters. Now let's talk about the Mendel's experiment. All right, so it was done in 3 stages. Okay, first is he made sure that plant which he had chosen must be true breeding. All right, so this was very important. The plant that he chose was true breeding. All right, and how a true breeding plant is created or generated? By letting the plant to undergo self-pollination or self-fertilization. Okay, so this true breeding plants, also known as the, you can say, pure varieties, these are obtained by self-pollination. So there is no cross-contamination with the other pollens. Now what is true breeding? So true breeding refers to the phenomena where any cross is conducted, all right, produce offerings with identical phenotypes, implying that the organism is homozygous for every gene. Clear? So this is known as the true breeding. Okay, and they are produced by the self-pollination or self-fertilization. Okay, and here the offerings which is produced, they have the identical phenotypes. All right, and these are homozygous for every gene. Homozygous means, so if we take the character height, then homozygous for every gene means capital T, capital T. So this is homozygous dominant. Clear? So these are true breeding lines, or you can say pure breeding lines. Clear? Now, he performed the process of cross-pollination of alternate forms of trait. Alternate forms of trait means the contrasting characters. Now the resultant generation obtained was termed as the hybrid, and these hybrid formed are called as the F1 generation or the first filial generation. Clear? So what does this mean? He did the cross-pollination of alternate form of traits. Again, if we take the example of the height, then what he did, he crossed the alternate form of trait. So one was the homozygous dominant, he crossed it with the homozygous recessive. Clear? So this is homozygous dominant, and this is homozygous recessive. Okay, this is clear. And then the F1 produced here will be T and small t. Okay, this is the, you can say, hybrid or the F1 which is produced. Clear? Here you can see how this T and small t hybrid is produced, because here T, so this is the gamete produced here, and small t is the gamete produced by this parent. So suppose this is the male parent, this one is the female parent. So male parent will produce gamete capital T and female parent will produce the gamete small t, and the hybrid will have the combination of these two. Clear? And this is known as the F1 or the first filial generation. Clear? Next is he allowed the hybrid to self-pollinate up to 5 generations, and these generations are subsequently termed as, so first generation was F1. All right. Now F1 was, there was selfing of the F1. Okay, so we can write there was the selfing of the F1, and selfing of the F1 will produce the F2, and again selfing of the F2 will produce F3, and so on. After F3, F4. Clear? So on. Clear? And because of the selfing, I've already told you, true breeding varieties are produced. Clear? Now let's study about the results of the Mendel experiment. What were the results? Okay. Now when the self-pollination was made and the F1 generation was obtained, it was found that the resultant generation would express only one of the trait and not the other. So the recessive trait was not expressed. Okay, so this is the trait which is not expressed is known as the recessive trait. Clear? And here the trait which was expressed is known as the dominant trait. So the trait which is being expressed is called as the dominant trait, whereas the one which is not expressed is called as the recessive trait. Clear? Now in the F1 generation, which was obtained by the self-pollination, the dominant and the recessive traits obtained were in the ratio of 3:1. Clear? So in the F2 generation, 75% of the offerings which appeared in the F2 generation had the dominant trait, while the 25% had the recessive trait. Clear? So initially F1 was the hybrid. Then this F1 was self-pollinated. I've already told you F1 was self-pollinated, and after self-pollination of the F1, F2 is obtained and 3:1 ratio was obtained. So 3:1 is the phenotypic ratio. Okay, so this is the phenotypic ratio which is observed in the monohybrid cross. Clear? Monohybrid cross, you know, it is a cross involving one pair of contrasting character. Okay, so here only one pair of contrasting character is involved. Clear? Okay, again if you talk about the height only, so you can say capital T cross with small t. So here only one character, height, okay, is taken in the monohybrid cross. Clear? Here hybrid will be, or F1 generation will be small t and small t. So this is the F1. Clear? Then selfing of F1 is done. Clear? And a ratio of 3:1 is obtained in the F2. We'll talk about this, how 3:1 is obtained in the F2. Clear? So here you can see. So these were the parent, capital T, small t and small t. So this is a monohybrid cross. Okay, this is a monohybrid cross. Okay, so this is the tall parent and this one is the dwarf parent. Clear? So these are the gametes that are produced. All right, so here we draw a Punnett square. So this is known as the Punnett square. Okay, so capital T, suppose this was the male parent and this one is the female parent. So capital T, these are the male gametes and these one are the female gametes. So here you can see, if we do a cross here, so this is the genotype of the progeny. Clear? So here you can see in F1 all are tall. Okay, it has the capital T. This also has capital T. This one also capital T. All right, and this is the small t here. Clear? So here if you see, if you do a cross like this, capital T cross with small t and small t, gametes produced will be T and small t. Clear? So now the F1 will be T and small t. So this is the F1. Now selfing of this F1 means capital T small t is crossed with capital T and small t. So this is known as the selfing of the F1. Okay, selfing of F1 is being done here. So here what will be the gametes? T and small t and T and small t. These are the gametes produced. This will produce T and another small t. This will also produce T and small t. Clear? So now we draw a Punnett square. So here capital T, small t, capital T, small t and small t and small t. So this is what? F2 generation. Clear? Now you can see this one will be the dominant, this one will be tall. This will be tall as well. This will be also tall. And this one will be dwarf. Clear? So what is the ratio here obtained? Ratio here obtained is 3:1. So 3 are tall and 1 is dwarf. Clear? And this is the phenotypic ratio. Okay, this is the phenotypic ratio. 3 are tall and 1 is dwarf. Clear? Now, this was the phenotypic ratio. There is one more ratio, genotypic ratio. So what will be the genotypic ratio here? Again, let's understand by this. So we had capital T cross with small t and small t. Here capital T small t, this is the F1 or the hybrid. Then the selfing is done, capital T small t. It will be crossed with capital T and small t. Then again you can draw the gametes here. And by using the Punnett square, you can tell about the genotype of the progeny. This will be capital T, capital T, small t, capital T, small t and small t and small t. Okay, so what is the genotype here? Genotypic ratio. So one is this one. This is homozygous dominant. This is homozygous heterozygous dominant. This is also heterozygous dominant. And this one is homozygous recessive. Clear? So this is homozygous recessive. Okay, this one is heterozygous dominant. Okay, then this one is homozygous dominant. Clear? So what is the genotypic ratio here? Okay, what will be the genotypic ratio? So genotypic ratio will be 1:2:1. Okay, 1 is homozygous dominant, so this is HD, homozygous dominant. Then we have 2 of the heterozygous dominant. So I'm writing heterozygous dominant, and the third one will be homozygous recessive, HR. Okay, so this is HR, this is HETD, okay, this is HD. Alright, so this is how you get the genotypic ratio. Clear? So here you can see there is a selfing of the F1. So this one was the F1. Okay, this is also F1, so there is a selfing of the F1, and what you get this. Okay, so here in the F2 you will get the 3:1 ratio. Clear? 3 dominant and 1 okay, 3:1 ratio is obtained. This should be small t. Clear? Small t and small t. Now the capital T, this is homozygous tall, capital T, small t, this is heterozygous tall, capital T and small t, this will be heterozygous dwarf. Clear? Now genotypic ratio, I've already told you, it is 1:2:1. Clear? So 1 will be the homozygous tall individual, 2 will be the heterozygous tall individual, and 1 will be the homozygous dwarfed individual. Clear? Now Mendel further found that the phenotypic ratio of 3:1 of dominant to recessive form of a trait was actually a genotypic ratio of 1:1 of pure dominant, hybrid, and the pure recessive forms. So this we have already discussed. 1 was the homozygous dominant, means the pure dominant. Then 2 ratio was for the hybrid, okay, heterozygous. And the 1 ratio was for the recessive one, homozygous recessive. Clear? Now the traits which remain hidden in the F1 generation got expressed in the F2 generation. Clear? We have seen if the F1 was T and small t. So here the phenotype was tall. So the recessive phenotype, okay, it was masked here by the dominant gene. It was not shown in the F1 generation, but in F2 generation you can see we got 3:1 phenotypic ratio, okay, and 1 was the dwarf. So the recessive character, it appeared in the F2 generation, and 3 were tall. Okay, that is what the meaning of this line. The traits which were hidden in the F1 generation, it got expressed in the F2 generation. Clear? When we got the ratio of 3:1. Now this was later on proved in the F3 generation also. Clear? Now reasons for the Mendel success. Why the Mendel got success here? Because he selected the true breeding or the pure lines, okay, pea plant for his experiment. So true breeding again means they were homozygous, okay, they were homozygous for a trait. Clear? And this homozygosity is achieved by self-fertilization or self-pollination. Next is he studied single trait at a time. Okay, that's why this was also the reason for the Mendel success. He was studying a single trait at a time. So he studied about the height first, okay, then he studied about the seed shape, then he studied about the flower color, and so on. Clear? Now next is he kept an accurate mathematical record of his breeding experiment and noted down the number of each type of offerings produced in each cross. So this was very tedious job. Alright, so he did everything. So he used this mathematical procedures or mathematical system to record everything, and he also noted what is the number of the, you can say, the progeny produced. Okay, the number of each type of offerings. So whether the offering were tall, whether it was dwarfed, okay, or whether it was of the intermediate height. Okay, So they or whether it had that intermediate genotype. Clear? Okay. So all of these the Mendel observed, and he kept a mathematical record of that. Okay, and this was a very tedious job to keep record of all the offerings that are produced and what types of offerings are produced. Clear? Whether they were tall, and what was their genotype. Clear? Now next is, he was lucky enough to select the seen traits. Okay, as the gene for these traits are located on 4 different chromosomes. Clear? So again, he selected the trait that were easily observable. We can see the phenotype and we can tell. Okay, so they were well-defined characters, and the genes, they were located on the different chromosomes. This was also a great factor that contributed to Mendel's success. These genes, they were present on different chromosomes. So if the genes are present very closely to each other, then there are chances that they are inherited together. And this is known as the linkage, when the genes are present very close to each other and they can be present and they are present on the same chromosome. But here the genes, they were present on the different chromosomes. They were quite far away from each other also. Okay, so because of this, he can see the inheritance pattern. Clear? Alright, so this was about the Mendel's experiment. Hope you enjoyed this session. Okay, we'll talk about the rest of the topics in the next lecture. Thank you very much for listening.